We are picking up where we left off talking about proteins. We talked last time about albumin, and back up one more slide. We talked about the categories of plasma proteins. Now, again, keep in mind that these are the proteins found in your blood, in your plasma. And there are two general categories, the albumin proteins and the globulin proteins. Albumin is in a group by itself. Albumin makes up 60% of the total protein in the body. Because it is such a large group, it is categorized by itself. Okay, so that was the first group. We talked about albumin kind of being on its own. Now we are going to talk about all of the beta globulins. Okay, and the beta globulins can be broken down into these four groups. We're going to talk about all of the globulins in their own little groups. So our first globulin that we're going to talk about are the alpha-1 globulins. And most of alpha-1 globulins are alpha-1 antitrypsin, also known as alpha-1 antitrypsin, sometimes called AAT for alpha antitrypsin, alpha-1 antitrypsin. And 90% of all alpha-1 proteins are alpha-1 antitrypsin. Okay. And alpha-1 antitrypsin is what we call an acute phase reactant. Now, acute phase reactant is a phrase that we are going to use a lot. And what an acute phase reactant is it's something that is increased during particularly acute or chronic inflammation. So alpha-1 antitrypsin is a sensitive marker for inflammation. Now, inflammation can mean anything. Okay, inflammation can mean you have a severe infection. Inflammation can mean that you have an autoimmune disease. Inflammation can mean that you broke your leg. Okay, there's a lot of different reasons, or you exercised too hard, or you're on your period, right? There are a lot of reasons that inflammation can be increased inside of a human body. So inflammation is a very general term, but testing for inflammation, and we'll get more into this in later chapters, but testing for inflammation can be a really good starting point to figuring out if and what is wrong in a person. Okay, so our first acute phase reactant is alpha-1 antitrypsin. Okay, it's increased significantly during acute and chronic inflammation. Right? And the way that alpha-1 antitrypsin works is during its, I lost my train of thought. Alpha-1 antitrypsin, one of its main jobs is it is supposed to control this enzyme called elastase. Now, elastase is produced during infections, particularly infections. So when the body is infected, okay, that's late. When the body is infected with a virus or a bacteria, elastase will be produced in an attempt to get rid of the bacteria or the virus. Elastase is an enzyme that breaks down elastic components of which bacteria and viruses tend to be made out of. Okay, so elastase is an enzyme produced during infection. In an attempt to control the elastase, because just like everything else, too much of a good thing is a bad thing. So too much elastase is a bad thing. So something else your body produces during inflammation is this alpha-1 antitrypsin. And the alpha-1 antitrypsin basically neutralizes elastase, as you can kind of see here. The green one is the elastase in the picture, and the purple one is the alpha-1 antitrypsin. The alpha-1 antitrypsin is produced whenever elastase is produced to kind of neutralize it so that we don't have too much elastase. Okay. And so that is why alpha-1 antitrypsin is produced and increased during inflammation, because elastase is produced during inflammation. However, in alpha-1 antitrypsin deficiency, so with all of these proteins, we're going to talk a lot about what happens if there's too much of that protein or not enough. In alpha-1 antitrypsin deficiency, so a decrease in that alpha-1 antitrypsin, that is a genetic disease that it tends towards being common in Caucasians. So this is more common in white people than any other racial group. It is a genetic disease, and it's a genetic lack of alpha-1 antitrypsin. And so what can happen in alpha-1 antitrypsin deficiency is basically the elastase goes unchecked. If there is no alpha-1 antitrypsin to neutralize it, the elastase just hangs around. Okay, and eventually what will happen is elastase is meant to attack or break down any of the stretchy fibers in our body, specifically elastin. Okay, elastin is a protein that is very common in collagen and a lot of the other tissue fibers that make up the stretchy parts of our body. A main component of that is our lungs, right? Think about what your lungs have to do. They get bigger and they contract, right? They're elastic. They're part of the most elastic parts of our body. They get bigger and they contract thousands of times a day. And if they lose that elasticity, they don't work. Being able to blow up like a balloon and sink back down is a big part of how your lungs work. Without that, your lungs would not be able to gather oxygen and then push out carbon dioxide like it's supposed to. So in alpha-1 antitrypsin deficiency, this is a relatively common cause of lung disease. It can also cause liver disease because there is elastin in your liver. There's elastin in other places too. The uterus has a lot of elastin, vaginal canals have lots of elastin. But the thing it's most known for causing is, I don't care about you, Siri, sorry, is lung disease. Okay, so alpha-1 antitrypsin is a genetic lung disease most common in Caucasian populations. And it is actually, I don't have this written down because you don't have to memorize these numbers, but alpha-1 antitrypsin deficiency is one of the most common genetically lethal diseases in Caucasians. Caucasian populations tend to have fewer of these genetically lethal diseases than other populations. But this is the most common one. Okay, and eventually it will cause permanent lung damage and the lungs will no longer work. Right, and then our two other alpha-1 globulins, remember that alpha-1 antitrypsin makes up about 90% of all of those alpha-1 globulins, but we also have alpha-1 acid glycoprotein and alpha-1 anti-chymotrypsin, both of which are acute phase reactants. And that's all we're going to say about them. They have other jobs, but again, they're mostly increased during inflammation. Okay, and that's all we're really going to talk about with them. All right. Another alpha-1 globulin that we have, and the other one that we're going to talk about kind of in detail, is alpha-fetoprotein. An alpha-fetoprotein is a very specific protein. It's an alpha-1 globulin that is secreted by the yolk sac and liver of a growing fetus. So alpha-1 or alpha-fetoprotein, as you can probably guess from the name, right? It's got fetus right in the name. It is not typically produced in non-pregnant adult humans. Alpha-fetoprotein is typically only secreted by the yolk sac and liver of a growing fetus. So a growing fetus will produce alpha-1 fetoprotein, and alpha-fetoprotein will appear in the gestational carrier serum around 16 to 18 weeks of gestation during pregnancy. So again, typically, I don't know why this is being weird. There we go. Typically during a non-pregnant adult will not produce alpha-fetoprotein. Now, alpha-fetoprotein is a sensitive indicator of the health of the fetus, especially when it comes to brain development. You will see an increase And this is kind of weird. You usually think of like a genetic disease being a decrease. This is one of the few where you see an increase. So in neural tube defects, things like spina bifida and anencephaly, if you've never taken any kind of developmental class or talked about this, the way that a developing fetus grows is the neural tube, what becomes the spinal column and the brain, starts out as a flat sheet. And eventually that thing closes over and forms a tube. So it starts out as a flat sheet of cells and it folds over, and that tube becomes the spinal column and the brain. However, if that tube doesn't close on the ends, it's supposed to be a completely closed tube. If it doesn't close on either end, it's called a neural tube defect. If it doesn't close at the top where the brain is, it's called anencephaly. The fetus doesn't have a brain. If it doesn't close at the bottom, it is something called spina bifida, which usually results in part of the nervous system being outside the body because it wasn't closed in properly, which can lead to death, but it can also lead to relatively minor physical disabilities. I don't want to judge people's disabilities, but relatively minor, meaning that it's survivable. But their legs may have decreased function and things like that. But that is what a neural tube defect is. It's when it doesn't close on either end. This is what is typically caused by folic acid deficiency. So that's why most prenatal vitamins have tons and tons of folic acid in them, because folic acid has been proven to decrease neural tube defects. But in a fetus that has a neural tube defect, there will be an increase in alpha-fetoprotein, and that increase can be detected in the blood of the gestational carrier. So there's a lot of really cool genetic testing going on where you can have your blood drawn. If you are a person that is carrying a baby, you can have your blood drawn at 16 weeks gestation and have all kinds of things tested on the fetus growing inside you, which is pretty cool. So you'll have an increase of alpha-fetoprotein in neural tube defects, and you'll have a decrease in things like Down syndrome and other trisomies or other chromosomal abnormalities. And it can also be found in the serum of non-pregnant adults with ovarian or testicular cancer. So neural or sex chromosome or sex cell cancers can cause the abnormal production of alpha-fetoprotein. Because again, non-pregnant adults should not be producing alpha-fetoprotein at all. Okay, so that is the last of our alpha-1 globulins. The major ones, again, alpha-1 antitrypsin is our biggest one, and then other acute phase reactants, and then this alpha-fetoprotein. Next, we have our alpha-2 globulins, and those are the three that we're going to talk about. I know there's a lot on these screens. Unfortunately, this is kind of one of those places where you just got to memorize stuff, which isn't very fun and it's a little boring, but it is important for you to know what all of these proteins do individually. So alpha-2 globulins, the first one we have is haptoglobin. And notice again that haptoglobin is an acute phase reactant. It is increased during inflammation. But haptoglobin's main job is to bind free hemoglobin to be recycled by the spleen. So in the normal course of being alive, hemoglobin is supposed to be inside your red blood cells, right? It's what carries oxygen through your red blood cells. Your red blood cells are going to be recycled. They're going to be destroyed. An average red blood cell lasts about 120 days, just about 3 months, and it will be destroyed. And that hemoglobin protein is actually recycled. So it is very normal for you to have free hemoglobin in your blood, a very small amount, due to the recycling and destruction of older red blood cells. Those old red blood cells break down, and that hemoglobin is grabbed up by the haptoglobin and taken to the spleen where new red blood cells are made. That's normal. You can see a decrease of haptoglobin during hemolysis. Now, hemolysis is the breakdown of red blood cells. Both intravascular and extravascular. If you don't know what those words mean, it doesn't matter right at this moment. We'll talk more about it later. If you've taken hematology or you're in blood bank, you may have heard those words already, which is why I specified. But if you don't know what those words mean, it's fine. It just happens in hemolysis. Again, the breakdown of red blood cells. So the reason that that causes a decrease of haptoglobin is because the breakdown of red blood cells, the unnatural breakdown of red blood cells, causes an excess of free hemoglobin. When there's more free hemoglobin, that haptoglobin will get bound up with the free hemoglobin. The more free hemoglobin there is, the more haptoglobin is going to get bound up with that free hemoglobin, and less haptoglobin will be free in the serum. Okay, so you see a decrease, and I guess maybe we should specify. It's a decrease in unbound haptoglobin. Because remember again here that we're looking at plasma proteins. So these are proteins in the plasma, proteins floating around in the bloodstream. So you see a decrease in unbound or plasma haptoglobin when there is increased free hemoglobin, because that haptoglobin gets bound up with the hemoglobin and pulled out of the blood. So what you're measuring in the blood becomes decreased. And hemolysis happens during a lot of different things, but specifically transfusion reactions. So if you are transfused the incorrect type of blood, your body will destroy it all because it's not the right kind. Your immune system says, this doesn't belong here, and it tries to get rid of all of it. You can also have a hemolytic anemia, which is a type of genetic disease. There's a lot of them. Sickle cell anemia is what we've talked about. That's a hemolytic anemia. It causes the breakdown of your red blood cells. And then something called BIC, which again is just something that causes the breakdown of red blood cells. Okay, so that is where you will see a decrease in unbound or free or plasma haptoglobin, is when there is hemolysis. And you'll see an increase of haptoglobin during inflammation because it is an acute phase reactant. It is produced when there is inflammation. So you'll see an increase of it. Infection, trauma, all of the things we said earlier about the various causes of inflammation. Okay. Our next protein is called ceruloplasmin. And ceruloplasmin is a protein whose job is to carry and transport and manage copper. Okay, so the ceruloplasmin is a protein that contains copper, and this is how your body maintains safe copper levels inside your body, because copper is a heavy metal. It is absolutely essential. Our body needs it for various things, but it is highly toxic, like most heavy metals. Okay, things like lead and mercury and copper, right? Those are things that are all necessary, but highly toxic if too high. So ceruloplasmin is what is responsible for kind of maintaining the free copper in your body. Okay, it binds up all that copper. It gives the copper a place to go. It gives all the copper a home. And it kind of neutralizes all of that toxic copper. Now you will have a decrease in ceruloplasmin in something called Wilson's disease. And Wilson's disease is a genetic disease where you don't produce enough or any ceruloplasmin, depending on how serious your disease is. And what will happen is if there are fewer ceruloplasmin molecules or proteins or none, the copper in your body will just be free. Okay, it doesn't have a place to go. There's nobody managing it. It doesn't have a nice little house to go into. And so the copper in your body just roams free. And because it's just roaming free, it's incredibly toxic. Your body will try to shove it in any place that it can. It will lead to increased free serum copper, and it will eventually build up in the organs as your body tries to deal with it. It'll build up in the kidney, liver, brain, and it can eventually cause kidney and liver failure and neurological damage. So it's an incredibly serious disease, excuse me. And one of the kind of hallmark signs of Wilson's disease, and if you ever take an ASCP exam, they freaking love this question, okay? The Kaiser-Fleischer rings, they love that question on the BOC exam. Not guaranteed that you're gonna see it, but it's very, very common. The Kaiser-Fleischer rings are the first sign of Wilson's disease. And if any of you ever watched House, there was an episode of House about somebody who had Wilson's disease. And the Kaiser-Fleischer rings were the first things that they looked for. And what Kaiser-Fleischer rings are these things over here. And it is basically a deposit of copper in the iris of your eye. And so there will be a ring, a brown, coppery colored ring around the iris of a person who has Wilson's disease. And that's often the first place that you see them. Okay, so if you ever have a question that has Kaiser-Fleischer rings in it, you don't need any other information. The answer is Wilson's disease. Okay, a decrease in ceruloplasmin, genetic decrease in ceruloplasmin. Okay, and then our last one is alpha-2 macroglobulin. And alpha-2 macroglobulin is really important because it is the largest plasma protein by size. It is a protease inhibitor similar to AAT, which is alpha-1 antitrypsin, and so it does serve a similar function to alpha-1 antitrypsin, but it is an incredibly important protein when it comes to nephrotic syndrome. Because we talked about nephrotic syndrome back here, we talked about albumin, that when kidneys are damaged, particularly with nephrotic syndrome, albumin and other smaller proteins will be lost through the kidneys. All the proteins will fall out of your plasma and go out into your urine, which is not normal. Okay? But alpha-2 macroglobulin, because it is so large, is not lost through that diseased kidney membrane. So when a person has nephrotic syndrome, all of the other proteins are going to be lost, specifically albumin and any other smaller proteins will be lost through that damaged kidney membrane. But the alpha-2 macroglobulin is too big because it's the largest, and it will not be lost as easily. And because it is not lost during nephrotic syndrome, you will have an increase just because all of the other proteins are being lost, but your body will also produce more alpha-2 macroglobulin to make up for the loss of the other proteins. Okay, the glomerular membrane, if you don't know that name, that's the kidney membrane. I know we haven't talked about kidneys yet, but we will. Okay. All right, so next we have our beta globulins. So those are our alpha-2s. Next we have our beta globulins. And our major protein component of the beta globulins is transferrin. And transferrin is the protein that's responsible for transporting iron from the intestines to the bone marrow or other storage sites. So iron is incredibly important, specifically in your red blood cells. It's important for carrying oxygen. It's important for making hemoglobin and forming your red blood cells. And the major carrier protein for iron is transferrin. Okay, remember that iron, like the chemical name for iron is Fe, and it also called like ferrous or ferric. That's not how you spell that. Okay. So iron, even though we say it iron in English, In Latin, it's ferric, ferrous, Fe. So transferrin is iron. And the way we actually measure transferrin in our body is not actually measuring the amount of transferrin. That's typically not super diagnostically relevant. The biggest thing that we look for when we're looking at transferrin is how saturated that transferrin is. Because that transferrin is responsible for, again, moving iron from place to place, where we need it versus where we get it from. And so when we're looking at transferrin saturation, we're looking at the iron levels in a person. So transferrin saturation measurements are used to monitor and diagnose iron levels. Okay, if you have decreased saturation, that means fewer of your transferrin molecules have iron on them. So you have a decrease in iron. Okay, so you will have an increase in saturation. An increase in transferrin saturation means you have more iron than normal. So you'll have an increase in saturation in something called hemochromatosis, which is just a hereditary iron overload. It's a genetic condition where you produce too much iron, or you overload your body with iron. And a person who has hereditary hemochromatosis will also usually have increased production of red blood cells, which again doesn't sound like it would be a problem, right? How can you have too many red blood cells or too many oxygens, right? Those are good things. You can't have too many. That's not correct. Everything, almost everything, within a human body needs to be maintained in a certain range. Too much of something can be a bad thing. In the case of red blood cells, too many red blood cells means your blood's too thick. Your blood pressure will be too high. Your blood will basically get caught in all of your tiny, delicate little capillaries and will cause organ damage. Not a good thing. So there's actually a really interesting, it's not even that interesting, but kind of a cool thing to think about. Treatment for hemochromatosis. Can anybody think of what it might be? What do you do if you have too many red blood cells?
You bleed yourself.
You remove them. Yeah, it's called therapeutic phlebotomy. It's a fancy word for it, but it basically just means you donate blood.
I only knew that because I met someone who had it before.
Yeah, there you go. It's not terribly uncommon, but yeah, it's called therapeutic phlebotomy. You just have your red blood cells pulled off every couple of weeks so that your hemoglobin stays in the correct range. There's talks of, and it's hard to know these types of things, but bleeding people, right? Their bleeding used to be a common treatment for all kinds of stuff, right? You were sick, well, we'll just bleed you, bloodlet you, we'll just cut you open. And it's entirely possible that when they did this, that there were times that people were actually cured or felt better after they were bled. Part of it may have been some of them had hemochromatosis. There's other conditions that kind of go along that same lines, but hemochromatosis, you have an increase in transferrin saturation because you have more iron than you're supposed to. And then you have a decrease in saturation in iron deficiency anemia, right? A decrease in iron means that you're going to have a decrease in transferrin saturation. Okay. And then transferrin is actually what we call a negative APR, APR being an acute phase reactant. It is actually the opposite of all of those acute phase reactants we talked about. It is decreased during inflammation. So it's a negative APR. It is decreased during inflammation. Hemopexin is a similar protein to haptoglobin. It's not exactly the same, but it is similar. It removes free heme from circulation. Again, similar to haptoglobin. And it is kind of similar to haptoglobin. It is increased during inflammation and decreased during hemolytic diseases. Very similar like what we talked about with haptoglobin. Okay, next we have our beta-2 lipoproteins, which are the major components of LDLs. That's all we're going to talk about with those. We talked about them before. We talked about lipoproteins. Okay, beta-2 microglobulin is a cell membrane protein on most nucleated cells. But there are particular reasons, or for reasons that you don't particularly have to know, there are very, very high levels of beta-2 microglobulin on lymphocytic cells, which are white blood cells. WBCs, white blood cells. So when you have specifically cancers, neoplasm is just a fancy word for cancer, if you didn't know that. But it can be increased in neoplasms, especially those that involve lymphocytes. So lymphomas and leukemias and things like that, that are cancers of the white blood cells, you can have a specific increase of beta-2 microglobulins. And then our last one is a protein called C-reactive protein, also known as CRP. And if you've ever worked in a hospital or a lab, or you've had your blood drawn for any kind of stuff, this is a very, very common test to test for inflammation because it's a highly sensitive. CRP is one of the first things that's going to start going up when a person has inflammation. However, it is non-specific, meaning that it is one of the first things to go up, but it's going to start going up for any kind of inflammation. For the most part, it is not going to specify, do you have an infection or inflammation due to a tissue injury or whatever else, okay? So it is highly sensitive, but very nonspecific. If your CRP is increasing, that means that you have some kind of inflammation. That could be useful, it could not be useful. However, CRP can be specific in one specific case, that it is increased in a myocardial infarction. And that's a fancy word for what? Who knows what that means? A heart attack. Myocardial infarction is a fancy word for a heart attack. So you can see a severe increase in C-reactive protein during heart attacks. Now, heart attacks are death of heart muscle tissue and inflammation. So it is a type of inflammation. But C-reactive protein in particular is a very sensitive marker for heart attacks. All right. So those are our beta globulins. Beta globulins. Next we have our gamma globulins. That word is really hard. Gamma globulins. Gamma globulins are also known as immunoglobulins or antibodies. Those words all mean the same thing, gamma globulin, immunoglobulin, antibodies. And they're highly specific proteins assembled to recognize and destroy foreign antigens. They are the main component of our immune system. So they increase specifically when there is a foreign antigen in our body. Now, foreign antigens can mean infection, and a lot of times they do, right? A viral or a bacterial infection. It can also mean you received a vaccine. It can also mean that you received a blood transfusion that that's the wrong blood type. It can mean a lot of different things, but they're meant to attack and destroy foreign antigens. I think of them as like the police force of the human body. They're the ones that are going around, right? And I know I'm just getting, every year this joke, and it's not a joke, but this lands less and less every year. But has anybody ever seen the movie Osmosis Jones? Yes. Okay, so Osmosis Jones, when I was a kid, right, a million years ago, Osmosis Jones was the movie that they made us watch on movie days in school, right? It was always Osmosis Jones. And that's because it was vaguely scientific, and so they were like, oh, right, it's a cartoon movie and it's vaguely scientific, so we can watch it in school. But Osmosis Jones is about a white blood cell. okay? And the main character, Osmosis Jones, is a white blood cell, and he's a cop in the police force that is the inside of this human body. And he's searching down, a virus, whatever. Anyway. So, I think it's a funny movie. I don't know how well it holds up. I haven't seen it in a really long time. But it's a very cheesy movie. There's a really gross scene with a pimple popping. It's the whole thing. But your gamma globulins are your antibodies. They are, again, they are assembled to recognize and destroy foreign antigens. And they are specifically produced by plasma cells, which are also known as B lymphocytes. Okay, these are white blood cells. All of our other proteins, specifically albumin, but everything else too, where are those mostly produced? Where is albumin produced? The liver. Most of your proteins are produced in your liver. So it's important to note that gamma globulins are kind of unusual because they are not produced in the liver. They are one of the few proteins that are produced, plasma proteins, that are produced by plasma cells or white blood cells, not the liver. Okay. And there are 5 types: IgG, IgM, IgA, IgD, and IgE. You don't have to memorize what they do for this class. You may have to for other classes. But for this class, you don't have to know that. And you will see an increase in these gamma globulins whenever you have an exposure to a non-self antigen. Sorry. Again, because these are specifically created to recognize and destroy non-self antigens, when you have an increase in non-self antigens, you will have an increase in these gamma globulins because they are being produced to destroy whatever has shown up. This mostly happens because of infection, but it can also happen during allergies, right? Allergies are pollens or whatever other allergen that you have getting into your body. Pregnancy, right? We don't like to think about growing fetuses that way, but they are a non-self foreign body inside of our body. It can also be increased in autoimmune responses, so when your body is responding to your own cells in an inappropriate way. And also multiple myeloma, which is a cancer of plasma cells. So if you're producing too many plasma cells because you have cancer of them, you are also going to have an increase in these gamma globulins. And you'll have a decrease in these gamma globulins in hereditary deficiencies. So there are deficiencies of the immune system, right? Again, with the old movies. There's a couple of old movies. There was one that came out in, I want to say the late 80s, early 90s, but it had John Travolta in it, and it was called like The Boy in the Bubble or something like that. And he had to live in a bubble his whole life because he had no immune system. He had a genetic hereditary deficiency, which caused him to not produce any gamma globulins. And this is a very real thing. Some people only don't produce certain kinds. Some people don't have an absolute deficiency. But hereditary deficiencies will cause you to have a decrease in these gamma globulins. HIV and AIDS, specifically AIDS. So HIV is a virus that destroys your lymphocytes. That's what it does. So HIV is a very serious disease virus that systematically destroys your immune system from the inside out over decades. HIV is basically a Trojan horse. It gets into your immune system, it gets carried there. It gets inside your immune system and stays hidden for decades and decades and decades while systematically killing off everything that makes immune cells in your body. So eventually, when I don't know why I hate that term, but the term full-blown AIDS, I think is a really weird way to put that. But HIV progresses to an autoimmune deficiency syndrome, which is what AIDS stands for. And eventually you will have no plasma cells left. You have no immune systems left. Immune cells left. If you have no immune cells left, you cannot produce gamma globulins. So in the late stages of HIV and AIDS, you have no immune system, you do not produce any gamma globulins, and usually people with advanced AIDS will die of a secondary infection because they have no immune system to fight it off. And then anytime you have a protein losing state, like nephrotic syndrome, and we'll talk about some other ones, gamma globulins are relatively small, and they are lost across that glomerular membrane very, very easily. All right. Now, I know this slide looks really intimidating, but there's just a lot of examples on this slide, but hopefully a lot of them kind of make sense together and you don't have to memorize every single thing on this list. It's not about memorizing what's on this list.
We don't have that.
You guys don't have this slide?
No.
We don't.
Okay.
Not at all.
All right. Well, that's annoying. Okay. I will post it right after this. But we're just going to talk about it. You guys can just take notes on your other slide, I guess. I will post it. If I don't post it by I have class until 12:30. If I don't post this by 1 p.m., somebody email me and remind me, okay? Because it's entirely possible I'll forget. And I want you to make sure you have it, okay? But yeah, we'll just talk about it and then I will post it later. Okay? So this is hyper and hypoproteinemia in general. So again, remember, emia means in the blood, hyper means high, hypo means low. Hyperproteinemia means high protein in the blood. Okay, and you can have hyperproteinemia for two major reasons. The first one is basically, remember that concentration is a look at the amount of solutes versus the amount of solution. There's two ways to change the concentration of something. You can change the number of solutes, or you can change the amount of the solution. Okay, so proteinemia, or protein, we're looking at the concentration of protein in the blood. Again, there's two ways to change that concentration. You can increase or decrease the amount of protein, or you can increase or decrease the amount of blood, of plasma. So hyperproteinemia, one of the ways that you can increase the amount of protein, or I guess we should say increase the concentration, is to decrease the amount of water or blood or solution that they're floating in. Okay, and that is called hemoconcentration or dehydration. You're removing the solution, which means that relatively there's more stuff in the blood than there was before. And all of these are just examples of ways that you can be dehydrated, right? Inadequate intake of water, that's the easiest one. Excessive sweating, vomiting. Okay, if you've been vomiting for 4 days, you're gonna be severely dehydrated. Salt losing syndromes, right? Those are ones that your electrolytes are imbalanced and you're gonna lose water along with salt. Diarrhea, again with being sick. I know you don't know what Addison's disease is yet. It's fine. It causes dehydration. That's just an example of dehydration, huh? Yes. And diabetic ketoacidosis. Again, diabetic ketoacidosis or just type 2 diabetes, one of the major symptoms of type 2 diabetes is excessive urination, dehydration. So you can either have an increase in a decrease in blood volume, dehydration, or you can have an increase in the proteins. Now, the only way that this really happens, you don't typically very often have an increase in albumin or anything else. You don't have a natural increase in albumin very often. There are not really many diseases that cause you to overproduce albumin. But you can have an increase in globulins. Now, globulins, multiple myeloma, remember this is cancer of plasma cells. So if you have cancer of your plasma cells, those plasma cells are going to overproduce gamma globulins, which will cause an increase in the overall level of protein. There is a genetic disease called Waldenstrom's macroglobulinemia. Can you guess, based on this name, what this is an overproduction of? Beta 2 macroglobulin. right? We had a protein called beta-2 macroglobulin. Waldenstrom's macroglobulinemia is a genetic increase of beta-2 macroglobulin. You just produce too much. It's a genetic condition. Chronic inflammatory conditions, a lot of those globulins that we talked about, gamma globulins, alpha-1 antitrypsin, CRP, haptoglobin, lots of them, are acute phase reactants, meaning that they are increased during inflammation. If your body is in a constant state of inflammation because of these chronic inflammatory conditions that you are going to have a chronic increase in globulins because they are all increased during inflammation. I don't think that's correct. I copied this out of our book. And if this is correct, then I don't know why it's correct. So I'm copying this. I think HIV and AIDS is usually considered a decrease in globulins. I don't know why that would be there. I think that's a mistake. If it's not a mistake, then we don't have to know it for this class because I don't understand why that would be an increase in globulins. So sorry. Like I said, this is out of a chart in the book. So if I don't know why it's wrong, then somebody can explain it to me if you can figure it out. But I think that's a typo. Okay. So then we have hypoproteinemias. And again, hypoproteinemias, when we're talking about concentrations, you can either alter the amount of the substance or you can alter the amount of the stuff that it's floating in, the blood. So in hypoproteinemia, you can have an increase in the plasma water volume, also known as hemodilution, which is a relative hypoproteinemia because you're increasing the amount of water, which is diluting the concentration of the proteins. And this can happen from water intoxication, which is absolutely a real thing. There was a thing, it went around TikTok like 2 years ago, where a woman, I don't remember where it was. It was in the US for sure. I want to say it was in California, but I don't remember for sure. But she went to the beach all day, sat out in the sun, got really dehydrated, went home, and because she was really dehydrated and didn't want to get like heat poisoning or heat sickness, chugged like 8 bottles of water in like 4 minutes because she was so dehydrated. She chugged a bunch of water, and that actually causes water intoxication. She actually died. It's a very real thing. It causes all of the salt distribution in your body to go completely haywire, makes your kidneys fail, all kinds of crazy stuff happen. It can be really, really serious. So water intoxication is a very real thing. It is when you dilute out your blood to an extreme level. Salt retention syndromes can cause hypoproteinemia. Water, and again, we're going to get more into this later when we talk about acid-base balance, but water is going to follow salt. So when you lose salt, like over here, the water follows the salt out and causes dehydration. When you retain salt, your body tries to retain the water to dilute out the salt, but it can cause hemodilution as well. Massive IV infusions, if you're in the hospital and they're giving you saline or whatever else on repeat for days and days and days and days, again, you're getting too many fluids. Volume expanders, so for people who have certain conditions, they'll be on medication that causes them to retain water. So again, all of those things are related to an increase in water volume. You don't necessarily have to memorize each condition, you just have to memorize why it causes hypoproteinemia. Okay? And you can also have hypoproteinemia because of an increase in protein loss. Protein is being lost from your body. It can be lost from your kidneys in nephrotic syndrome, blood loss. If you're losing lots of blood, most of your protein is in your blood. So if you're losing blood, you're also losing protein. Burn patients have no skin, and they lose fluid and blood and proteins through their open wounds on their skin. Trauma, again, you're losing blood, you're losing protein. Inflammatory bowel disease. Inflammatory bowel disease often causes blood loss through the gastrointestinal tract. You're losing blood, you're losing protein. Okay. Can also have decreased protein due to decreased intake. You're simply not eating enough amino acids to produce the proteins that your body needs. Malnutrition, malabsorption, or starvation and anorexia. And then you can also have decreased synthesis. The major reasons of decreased synthesis are going to be liver disease and immunodeficiency disorders, because those are the two major places that proteins are formed, right? Most proteins are synthesized in the liver. If your liver is not working properly, it's not going to make proteins. And then immunodeficiency syndromes, our gamma globulins are produced in our plasma cells, which are immune system cells. If our immune system's not working, the plasma cells are not produced, and they do not produce gamma globulins. Okay. Again, I will post that. Okay, so now that we've talked about all of we obviously have not talked about all of the proteins in our body or in our blood, but those are the major ones we're going to talk about. They're the ones that are measured on a regular basis to test for certain conditions. And so now the question becomes, how do we test for proteins? How do we test usually for total protein and things like that? And the most common method for total protein measurement is called the Biuret method. And the Biuret method involves adding copper, a copper ion, and this copper ion actually binds to, or forms a complex with, I guess is a better word for it, but it binds to peptide bonds. So what this test is actually testing for is the number of peptide bonds, because that copper binds to the peptide bonds, and so it's the bonds in between the individual amino acids that are being counted in this method. Okay, and when this peptide bond copper complex forms, it changes from a light blue color to a deep purple color, which can then be measured by spectrophotometer. Again, this is the most common method for total protein measurement. It's easily automated and sensitive enough for clinical use. Interfering substances, like a lot of our other ones, are substances with strong colors, or strong substances that will affect the color and quality of the plasma. So hemolysis, remember, breaking open those red blood cells causes the plasma to turn dark red. Hyperbilirubinemia causes the plasma to turn like a bright orange color. And lipidemia, or lipemia, it causes the plasma to turn a white, milky color. So anything that's going to affect the color is going to affect the color change we see, right? You can imagine something that's changing from blue to purple. If you add in something that's really red into that mix, that's going to alter the purple color, and then it will make the results inaccurate. Okay. Now, another thing that can be really important is, remember that total protein, so the amount of protein in your blood is going to come in two categories. It's the albumin plus the globulin. Because all the proteins in your plasma can be broken into two categories. They're either albumin or they're a globulin. Okay, so if you have the amount of albumin and the amount of globulin, that should add together to the amount of total protein. Now, albumin is usually tested for by a bromocresol dye. You can either use a bromocresol green or purple, doesn't matter which. But when albumin binds to that dye, it forms a complex that changes colors. That's all you have to know about it. That can be measured by spectrophotometry, again. But these bromocresol dyes specifically bind to albumin, not all proteins. So those bromocresol dyes are specifically used for testing albumin. And again, this is a very accurate, sensitive, specific test. Has very few interfering substances. Globulin testing is not readily available, but globulin levels can be calculated. So when I told you that the only math that you'd be doing was total protein or was lipid math, I lied. You'll also have to do this math. Okay, it's really hard. I know it's really complicated. But globulins can be calculated by taking the amount of total protein and subtracting the amount of albumin, because total protein is albumin plus globulin. Subtract out the amount of albumin, you'll have the level of globulin. Okay? So globulin testing is not usually performed. Usually on a panel, a doctor will order a total protein and an albumin, and then globulin is calculated. The albumin-globulin ratio can be important in determining what is wrong with a person, right? So if a total protein is increased or decreased, you want to narrow down where the problem is. Is the problem that they have too much albumin, not enough albumin, too much globulin, not enough globulin? And what can kind of narrow that down is the albumin-globulin ratio. So once you have the albumin and the globulin, you can divide them to get the ratio. If you have an increased ratio, meaning you have more albumin than globulin, that indicates the underproduction of globulins. Genetic deficiencies, hypothyroidism, some leukemias, but you have a decrease in the globulins. A decrease in the ratio means you have an overproduction of globulins or an underproduction of albumin. All right, so those are the reference ranges you do have to know. There is the protein, total protein. That wasn't obvious. That's the total protein, albumin, and then the AG ratio that are the normal values. Okay. All right. So serum protein electrophoresis. This is a method that is often used when trying to figure out why somebody has an increase or a decrease in total protein or albumin globulin ratios. Because again, those individual proteins are not tested for super often. They can be, but if you have a patient that just has a hyperproteinemia or a hypoproteinemia and their AG ratio is off, that doesn't tell you exactly what's happening. That tells you that they're having a problem with their proteins, but there are so many proteins that it can be really hard to tell what's going on. So serum protein electrophoresis, again, this is a way of separating on a gel, and I think I confirmed that you guys have all seen this before, but it's a way of separating on a gel a person's protein. So you can take a sample of somebody's plasma that's been treated properly with all the right things in a dye, and then separate them on this gel. And the biggest proteins will be up here, and the smallest will be down here. And this also is complicated a little bit by the charge, because proteins don't always have the exact same amount of charge, right? But they will separate out like this. And it always goes, albumin is first, right? Albumin is the most, and it's going to be the darkest. There's going to be the most, and that one's going to be first. And then it's going to go in order. Remember, alpha, beta, gamma is the order in the Greek alphabet. Alpha is first, beta is second, gamma is third. So it's going to be the alpha 1, and then the alpha 2, and then the betas, and then the gammas. Okay, so it goes in order. Albumin, alpha 1, alpha 2, beta, gamma. Okay. Now, you don't have to memorize the relative numbers specifically. Like, I'm not going to ask you what percentage. You do have to memorize that albumin is the most, by far. Okay. Now, we can also take all of this data gotten by a gel and measuring the density of each of the bands, and we can put it onto something called an electropherogram. An electropherogram is just this data in a chart. So it's everything gotten from this gel and put in a chart. So this is the anode on this side, and this is the cathode on this side. Okay, So again, it's the exact same data, it's just put in a chart. And this chart is usually easier to look at than a gel, usually. Okay? And so this is what a normal electropharogram is going to look like. You'll notice the couple of things that you have to know is that the albumin spike is going to be by far the biggest, or it's supposed to be. There's by far more albumin than there is anything else. And then you have to memorize the order. Like I said, alpha 1, alpha 2, beta, gamma. The alpha, the beta, the two alphas and the beta and the gamma are all relatively short little bumps. This gamma one, this picture is not the best, but the gamma one is also typically considered a more broad peak, meaning that instead of being like a sharp peak like this one, it's kind of broad. It like drags out. I have a better picture in just a second. Okay, and these electropharograms can be really important in diagnosing specific protein deficiencies or abnormalities. And there are a couple of abnormal electropharograms that you do have to know, and they're all on this page. These are the only ones that you have to know. There are lots of them, okay? And if I do give them to you, I will give them to you in this specific format because it can be really hard on a piece of paper to try to figure out which one's bigger and which one's smaller. But I will give you ones where the things that have changed are highlighted. Okay, so I will always give it to you in this specific format. Okay. So this is a normal electropharogram. As you can tell, again, this one's a little bit better of a representation because this gamma one is kind of broad. Okay. There's a lot of gamma proteins, and that spike excuse me, is kind of broad and wide. We have albumin, alpha 1, alpha 2, beta, and then that kind of broad gamma one. That's normal. Okay, this one, where you have a specific increase in alpha 1 and alpha 2, is going to be increased in acute inflammation. Because most of the proteins, specifically alpha 1 antitrypsin, and a lot of the ones in the alpha 2 that we talked about are all what? What did we call them all? Acute phase reactants. They are increased in acute inflammation. So in acute inflammation, you will have an increase in those acute phase reactants, which will cause those peaks to be a little bit taller than they normally would be. Okay, a person who has alpha 1 antitrypsin deficiency will have either a little tiny bump in the alpha 1, or it has been known to be completely flat, like you'll see where it'll be like this, and it'll kind of skip that alpha 1. Okay, but that alpha 1 will be severely decreased in size, much, much smaller than the normal one. Now, liver cirrhosis. Okay. So what is happening in this liver cirrhosis? What do you have a decrease of?
Albumin.
Albumin, right? If you compare it to this one over here, there's a severe decrease in albumin. Why would you have a decrease in albumin in liver disease? Yeah, because albumin is produced in the liver. It's as simple as that. Albumin is produced in the liver. If somebody has a diseased liver, they're not going to produce albumin. Okay. And then you also have a pretty large increase in gamma globulins. And that's because gamma globulins are not produced in the liver. And remember that what is one of albumin's most important jobs? Its main function. What does it maintain? Osmotic pressure. Osmotic pressure is incredibly important. And when you're losing or decreasing the amount of albumin, your body is going to try to correct that as much as possible. And so it will start producing as many proteins as it can to maintain that osmotic pressure. So your body will up the amount of gamma being produced because it cannot control the amount of albumin being produced. The liver is failing, the liver's out. But gamma globulins are produced in plasma cells. They are not produced in the liver. Okay. This is a picture of multiple myeloma. Remember, multiple myeloma is a cancer of what? Plasma cells. Okay. This is a plasma cell cancer. So in multiple myeloma, you will have this very, very specifically increased and distinctive spike in the gamma globulins because there is a cancer that is overproducing that plasma cells, which will overproduce gamma globulins. This specific finding, this spike right here, is specifically called an M spike because it is almost completely diagnostic for multiple myeloma. You don't see anything like it except for in multiple myeloma. All right. And then nephrotic syndrome. We talked about nephrotic syndrome a lot. Nephrotic syndrome, again, the main symptom is going to be a decrease in albumin. Why? Why do we have a decrease in albumin? Is it because you're not producing as much?
No, it's lost in the kidneys.
Yeah, it's being lost through the glomerular membrane or through the kidneys. And so what your body will do when you're losing albumin is it's going to try to up the production of everything else. So in this case, because in liver cirrhosis or liver failure, your body is not capable of producing any of these proteins at a higher rate because these are all produced in the liver as well. But in this case, your liver is perfectly fine. It's your kidneys that are broken. So your body will overproduce the alpha-2s, the betas, and the gammas to try to increase the osmotic pressure again, to try to make up for the loss of albumin. All right. Okay, so I know we only have a few minutes, but I am going to try to kind of blast through. There's only like 3 slides here, so I want to try to get through as much of this as I can. I do still have 5 minutes. So I do want to talk briefly about enzymes. So we spent a whole chapter on proteins. Enzymes are just specific types of proteins that catalyze reactions. And hopefully this is not the first time you've heard of an enzyme. I'm sure they've talked about it in lots of other biology classes before now. But enzymes are protein catalysts. They're proteins that catalyze reactions. And their biggest thing is that they enable metabolic reactions to occur at body temperature. A lot of reactions that happen inside of our body don't happen on their own at body temperature. They might happen if you heated us up to a much higher temperature, but that's not something that can happen. So instead of heating us up, they've created these catalysts, which make things happen. And the biggest thing about catalysts, and I think I have another slide on it in just a minute, but they decrease the activation energy to allow reactions to occur at much faster rates. And these enzymes, again, it's really hard to overstate how important proteins and enzymes are. This includes muscle contractions, nerve conduction, respiration, digestion, nutrient degradation, growth, reproduction, body temperature maintenance, metabolism, on and on and on and on. Almost everything inside your body is controlled by some kind of enzyme, making some kind of reaction happen. Okay. And they are synthesized in the body under control of specific genes like all other proteins. So that's just kind of a generic thing. But how enzymes work, a lot of enzymes have what's called a lock and key mechanism, where the enzyme is specifically designed to have a 3-dimensional structure that fits a specific set of substrates. Okay, so this active site on the enzyme, its 3-dimensional structure is physically built to fit one specific thing. So enzymes are almost always incredibly specific. Most of the time, they only catalyze one reaction. Okay, they have one set of substrates that they are capable of catalyzing. Okay, and so the substrate will bind to the enzyme. That enzyme will cause some kind of change to happen, and then the products will leave. That's the most generic way to talk about enzymes. And some of the critical things about enzymes is that enzymes are not altered or consumed during the reaction they catalyze. Once enzymes are produced, they can catalyze the same reaction over and over and over again. They are not changed, they are not consumed, they do not need to be rebuilt. Only small amounts of enzymes are required because they are used over and over and over again. They are not consumed. And the thing is here is that they accelerate the speed at which a chemical reaction happens, but they do not alter the equilibrium constant, meaning that they don't alter the amount of product formed from a substrate. So if one substrate if one substrate is going to be, if the substrate is AB and the products are A plus B, meaning that you took that one thing and you broke it in half, right? If that's the reaction, it doesn't change the reaction to produce 2A plus B, right? It doesn't change the amount created. It only changes how fast that reaction happens. So it doesn't change the products or the substrates or the amounts of things that happen. It only changes how fast they happen. Okay? And this was kind of the main point, is that enzymes work by lowering the activation energy. The activation energy is the required amount of energy to make 1 mole of substrate formed from an activated complex. Okay, so it's the amount of energy required to make a reaction happen, basically. I think of it as like pushing a rock up a hill, right? So if you push a rock all the way up at the top of the hill, once you get it to the top of the hill, it's gonna roll down the other side, regardless of how much energy it took. If that's the reaction that we're trying to make happen, getting this rock to the top of the hill, all that's changing, I'm not changing me, I'm not changing the rock, I'm not changing how many rocks there are, I'm not changing how many rocks are gonna be on the end. All I'm changing is the size of the hill. It's changing the activation energy, the amount of energy required to make it happen. Okay, and I guess we're just not gonna get to that. Well, that's okay. Talk more about it after you take your exam on Wednesday.